Hello, everybody. Sorry for the delay. We had a problem with the Zoom and we couldn't manage to be connected, but it seems to be okay now. So we are going to start and try to keep time. So we are really pleased to welcome today the two researchers who belongs to the MOZAIC team at the Fresnel Institute. So Hervé and Thomas, in fact, are part of the MOZAIC team, which recently integrated the NeuroMarseille Institute as an associated team. And this team will participate to our research and training actions in the future. So the MOZAIC team is using and developing tools, photonic tools, to study life science and in particular neuroscience. So they are in fact an interdisciplinary group with physicists and biologists working together to design instruments for quantitative biology. So they have, in fact, a long-standing experience in developing these tools for developmental biology, immunology, but also neuroscience. And you will see all these kind of methods during their presentation. So I don't know if Hervé is here. Yes, hi Hervé. We cannot hear you. So we are going to have two presentations tonight. The first one will be given by Hervé and then the second by Thomas. So maybe we should start with the first one and then have questions and then go to the second one. So Hervé Rigneault will be our first speaker. Hervé is CNRS Research Director and is leading the MOZAIC group at the Fresnel Institute since 2000. He is currently developing multiphoton microscope and endoscope that aim to provide images in neuroscience. And I think today Hervé is going to present this work. The title of his presentation is Multiphoton endoscopy to study the brain activity in vivo on freely moving animals. So Hervé, the screen is yours. And okay. Again, we are sorry for the delay.
Yes, can you hear me?
Yeah, really well.
Very good. Okay. Okay. And then you should see full screen. No, not full screen. Sorry. Okay. So now once again. Okay. So you should see full screen. My screen. Is that right?
Yeah, that's right. So okay, as usual, if you have questions, you can use the Q&A system. Thank you.
Okay, so I cannot see it when I'm full screen, so I let you interrupt me to if there is any questions.
Okay, fine. I will let you know if we have questions during your presentation.
Okay. Okay, so thank you everyone for this introduction. So I'm Hervé Rigneault, and so today we will have two speakers on this talk. This is myself, Hervé Rigneault, and my colleague Thomas Chaigne. Will just follow my talk, and then the general title of this, let's say, quite peculiar talks, which is not really like in real biology, but more on development of technical tools, is what physics and optical engineering can bring to neuroscience. And so we are going to present two topics as it was presented on multiphoton endoscope and photoacoustic approaches. So just a few words to start with on the Institut Fresnel. I know we are all spread all around the Marseille area, but just to let you know where is the Institut Fresnel. So you see that this is the viewport down here, the first arrondissement of Marseille, and then Institut Fresnel is located on the campus of Saint-Jérôme. It's a laboratory that depends on the engineering department of the CNRS in CIS, and it has like main topics such as information, photonics, modeling in optics, nanophotonics, and also imaging. So we are, Thomas and myself, part of the imaging, let's say, department of the Fresnel Institute, and especially in the MOZAIC group. And this is a view of the Fresnel Institute. So Fresnel is 200 people among 100 PhD and postdoc and 100 permanent researchers. So that was for the short introduction about what is Institut Fresnel, and you're very welcome to come and visit us. Okay, so now I just jump in the topic that I want to address. I think I have something like 15 minutes, so I'll do my best to keep the time in here. I'm going to put my starter on, and there we go. Okay, so this is something that you know, right? This is the imaging the neuron activity patterns in vivo. And so this movie that I'm playing now is coming from the INMED, so the lab of my colleague Rosa Cossard, and she's studying the brain network activity. I don't want to go into any details on that, but the point is that there are assemblies of neurons that are flashing and that are flashing at sequential time that encode space or time. And this contrast mechanism is due to the fluorescence of the G camp that is actually a calcium indicator. And there is a wall field here in order to see and to understand the pattern of the brain. Right. So to do that, then what they do is that they use what they call a chronic glass window, which is an invasive surgery. It's actually to remove the bones and the part of the cortex in order to access the part of the brain which is interesting. And in that case, this is the hippocampus for that specific group. And this is the view of the intracranial cannula that they insert into the brain, and from which they can actually see the places that are deep inside. So most of the experiment, and I think all the experiments are actually performed in multiphoton microscopy, and this allows to have a good sectioning capability of the image. And they're performed on what they call a head restraints experiment. This is the case when the mouse has the head which is attached to the microscope, as you can see here, and then the bulky microscope objective is actually looking through this window at a few hundred nanometers just below the glass slide. Okay, so of course, this is quite interesting and lots of research which is done with those tools around the world, but there is a trend now to try to extend those tools to miniature devices in order to explore the brain activity, the network activity, but enabling then the mice or the rat or the monkey to move. And this is, for example, in the mice, for instance, through the one-photon devices, and this is a commercial device that is sold by the company Inscopix in the US. It's a tiny camera that can be set on the head of the mouse. And there is also a Chinese company, or let's say there was this nice paper in 2017, and probably a company that is going to release a 2-photon version of this device as well, as was demonstrated. So those devices, they are using green lenses, about 1 millimeter to 2 millimeter in diameter, and it just gives you a bit then the size of those green lenses when they go into the brain of the mouse, as compared to the depth that you can reach if you use 1-photon fluorescence, which is the case of the Incopix, or 2-photon fluorescence, or even 3-photon fluorescence. So you see that this is quite invasive. Right? So I'm just doing my research in that field. That is to say, can we actually do that through a fiber and try to miniaturize a bit the device? So it has to do to deliver short pulses, short laser pulses at the end of the fiber to collect the emitted photons and also to perform imaging. So I'll just close my window. There's noise outside. There we go. So what we are using here, it's holocore fibers. So holocore fibers are specific fibers, they call that specialty fibers, where the light can actually propagate in an air hole, which is in between a kind of structured material. And this peculiar property of the light to go through or to propagate in the air actually makes the interaction of light with the fiber very weak, such as that a short pulse remains short at the end of the fiber. So this is the type of fiber that we are developing together with our research colleague in Lille. The laboratory is called IRSICA. This is the CNRS French platform in order to develop novelty fibers or specialty fibers. And so just a few words on the fibers. So this is the place where the light is propagating in here at the center. And this ring, which is around here, is the ring that will collect the signal that will be backscattered. So the fluorescent signal. And what you see here, this is the double clad. This is what collects the light. And you will understand right afterwards how it works. And this is the core of the fiber. This is where we send the ultra short pulse. typically the pulse that will excite the G-camp reporter or any of your favorite reporter. So the transmission of this fiber is quite broad. You see between 800 nanometers up to 1 micron and 200 nanometers. And also it has a very low dispersion, which means that a short pulse at the entrance of the fiber, which is something like 2 meters, will remain short at the output of the fiber below 200 femtoseconds, and for every wavelength that we want to use to excite our fluorophores. So now a few words about the device, which is the scanner itself. So, right, so we want to make an imaging system which is tiny, which is small. So what we are using is a piezo scanner. So this cylinder, which is here, is a piezo tube. It's made from piezoelectric material that has the ability to change its shape when you put a voltage at the entrance. So actually, there are 4 cables because it is a 4-quadrant piezo system where you can actually scan along X and you can scan along the Y axis as well. But this, let's say, scanning range is very weak. It's of the orders of less than 10 micron. So in order to make an amplitude of the movement which is bigger, what we do is that we stick our fiber to this piezo tube and we use the resonant frequency of this freestanding piece of fiber, optical fibers that will shake. So this is our hollow core fiber, you know, the one I presented before, that is just glued to the piezo tube. And there is a micro lens system, which is then after that will image the fiber tip down to the sample into the head of the mouse, and then it will also back collect the fluorescent signal that will enter the fiber. And as you understood, it will be mostly collected through the double clad, you know, this ring I was presented you before, right? So this is just it's just like a confocal microscope, but it's a tiny system. And the scanning mechanism that we are using is actually not a raster scanning, you know, that is making zigzag in a conventional microscope. It's more like, in this case, this is a spiral. So it means that the tip of the endoscope will make an increased spiral from which we will excite the sample and we will collect the signal. So this is the resonant frequency of the piezo. So this was an earlier version. So now we are about 1000 hertz, 1 kilohertz. And then for the X, so this is when you shake this part of the fiber, this is, you can shake it 1000 times per second, right? And on the other side as well. And when you drive those 2 piezo with 2 voltages that increases with a phase shift between the 2, this is where you can make this spiral. So altogether, it's a small scanner, and that's a view of the scanner here when it's completely dismantled. You see here you have the piezo tube. This is the fiber optics, and as you understood, when you shake this one at the resonant frequency, then you shake the end of the fiber that makes the spiral, and this is the micro lens that is there that image the end of the fiber down to the sample. And this objective lens was there for calibration purposes, but this is the full system is only this kind of tube. And this is the design of the distal objective lens, which has, in this version, it has a 0.3 numerical aperture on the side, on the fiber side, and 0.45 on the sample side. So now our last version has 0.7 of numerical aperture. So hopefully we can collect enough signal. Right. Okay, so there is a little optical trick. I will go quick through that, but just to let you know a bit what are the problems that are challenging when you want to do this kind of devices, is the fact that the light, as I told you, is propagating into the hollow core of the fiber, and you want to image that core of the fiber down to the sample. And but this core is 20 microns, so you have a 20 micron spot here that you want to image down to a 1 micron spot. So if you do a demagnification of a factor of 20, you can certainly, let's say, change a 20 micron spot into a 1 micron spot. But the field of view would be also very limited. So in order to keep the field of view large, what we do is that we use another property, which is the property of a bead, of a silica bead, to focus the light just after the bead, such as when we stick one of those silica beads at the end of the hollow core fiber, what it does is that instead of having a mode diameter of 20 micron, Now the light is focused, and it's focused down 1 micron, such as now the magnification between this 1 micron spot and the sample can be only 1. And if it's 1, it means that when you shake the fiber for, let's say, 400 micron here, you can have a 400 micron field of view. So it's just a trick if you wish to increase the field of view, keeping the resolution of the device. So the whole view of the system is this one. So and
Hervé?
Yes.
I have one question. I'm not sure to understand why you need only one bead at the center.
Right.
Since you have many fibers around, is
right.
it necessary?
Okay. So sorry for the maybe I was not clear. Okay, so there is only one fiber core.
Okay, fine.
This device is just like one fiber, and there is only the central core that can actually guide the light through the sample. All this mesh which is around that you have told it's core, it's not core actually. It's just a kind of what we call a photonic crystal. It's a mesh of material that prevents the light to escape from this hollow core.
Okay.
So it's not so intuitive that the light can be guided into air. Usually in optical fibers, the light is guided in glass. But because of that mesh that prevents the light to escape from that tunnel, if you wish, this is the property. So again, this is only the core of the fiber, and we send the light only in the central part of the fiber. This is only to keep the light into the central part, right?
Okay.
Okay. So if you wish, at the end of the day, this is just like a single fiber.
Okay.
There is a single fiber core, and then so this is the reason we need to shake the fiber. We need to make a spiral with this fiber in order to change the location of this core in order to scan across the samples, such as you would do with a conventional single core fiber.
Okay.
It's a single core fiber, but it's a hollow core. And and the interest of the hollow core is that you can send short pulses inside to do 2-photon fluorescence. That's all the rationale behind.
Okay, thank you.
So thank you. So this is the view of the endoscope as it is now assembled. So you have the piezo, then the fiber and the lens, and it comes all together in these little sticks. So this was an early version. You will see later on then the new version. But at the time, so it was in 2018, it was like you see 7 centimeter long and about 4 millimeter in diameter. So you can use your laser in order to send into the endoscope. Then you collect the fluorescence, and that goes back into the detector. Or it can be 2-photon fluorescence and some other mechanism, just as second hormone degeneration to see collagen, or even the lipid using CARS. But for neuroscience, it would be mostly 2-photon fluorescence from the calcium reporter. So this is a closer view of the tip that is handheld in that case. So you see this little thing now here, and you find here a bit the details inside. So this is the piezo in red. This is the fiber. The fiber shakes, makes a spiral, and this is the micro lens that image the tip of the fiber down to the So the resolution of such an endoscope is about 0.8 micron in diameter, which makes a good resolution to see neuron. Actually, it's much less than a neuron. And in the Z direction, this is what you expect from 2-photon fluorescence. Yes.
Hervé, your sound is not really great. I don't understand why. Maybe it's because you are too close to the computer.
Okay.
I don't know. Sometimes we are missing some part of your. I don't know. Maybe we can try.
Okay. Is it? Can you hear me now?
Yeah, maybe it's better if you're not so close. Maybe.
Okay. Very good. Okay. So right. So I will stand at this distance from the screen. Sorry. So again, this is the resolution of the endoscope, which is about 0.8. So it's less than a micron in XY, and it's about 6 micron on the Z axis. So it gives a quite good resolution for such a tiny device. And that was an early example of what we could do in a tissue, for instance. So it is not related to neuroscience here. It's just a piece of tissue where you see this is the collagen from the rat tail or the mouse tail. And this is also in green, the collagen as seen by second hormone degeneration into a tissue that is coming from the hospital, as well as in red, these are the lipids that are present. It is seen using CARS, which is coherent antihistochroman scattering. It's a contrast mechanism that is able to see lipids. Okay, so now closer from the field of neuroscience, it's also possible to use the autofluorescence of the laser in order to make an image. So I think the field of view in here is something like 350 microns. This is the stratum corneum of the ear of a living mouse. And these other examples is the spinal cords. Maybe some of you know our colleague Franck de Barbieux, which is at ENT Marseille. And Franck is using mice with a window on the spinal cord. And you see here, so this is YFP tagged neuro axons. That's what we see here in blue. And the myelin sheet, which is seen by coherent Raman, is in magenta. So of course, our scope has been to develop and to push our technology to go into the brain. So very quickly, so we are now using novel type of fiber, but this is the same concept. There is a hollow core, there is a bead, and there is a double clad that collects the signal. So this is our latest versions of optical fibers that we are doing. So now our endoscope has a distal head of 2 millimeter diameter. And it was made indeed to go into the cannula that is used by our colleague at InMed in order to enter the endoscope into the head. So we are able now to do 8 frames per second. So this is an example of neurons that we can see in the brain. It was an ex vivo experiment on the brain slides. We have tried to do an in vivo experiment. So yet in this early experiment, the mouse has still the head which is attached. So it's just a demo to demonstrate that the endoscope can see neurons. And we were just entering our endoscope into the head of the mouse to see neurons. And indeed, we've been able to see neurons. But okay, so there has been the experiment had some problems, and we haven't been able to see the activity yet. So this is our next challenge. But for sure, we are able to see neurons. So at the time we did those experiments, we were able to do 1 frame per second. That was maybe a bit slow in order to see the activity of neurons. Hopefully now with 10 frames per second, we want to do that. So the scope of all this research is to develop this freely moving endoscope that can be attached to the head of a mouse. Using 930 or 920 nanometers femtosecond laser to detect GFP or GCAMP at 10 frames per second with let's say subcellular resolution with a field of view over 200 microns. So now we are actively developing what would be the endoscope brain interface. And actually, maybe some of you have followed the work from Cathy Ventalon at IBENS in École Normale in Paris. So Cathy Ventalon has developed together with the small company in Paris called Imetry, an endoscope brain interface that was done for the fiber bundle that Cathy Ventalon is using. So this is 1 photon fluorescence. So you understood that the endoscope that we are developing in Marseille is a 2-photon endoscope. And this is actually so it's very new work that we are doing now in order to design then the brain interface. So it will be altogether 35 millimeter. This part down here would be inserted into the head of the mouse. And there will be then an interface that is able to clamp the, let's say, endoscope part to the brain. And this kind of strange crown that you see here is in fact a spring, un ressort, in order to somehow connect very firmly the endoscope head to the head of the mouse. So it's kind of nice design in order to put the tension onto the device. It will be also possible to tune the Z depth in order to tune at which depth the endoscope is looking. So this will be done manually using this little screw that is there, this little ring that is there in order to do that. And okay, so we are trying also to develop, so in order to let's say, to disseminate this technology into the laboratories in Marseille, and I'm very happy if you want to come to me, if you come back to me in order to see whether we can make this device available to you. We are developing a kind of cart, a standalone equipment that will include a 2-photon laser for GFP. And so altogether, it looks like this now. So you see it's a kind of cart that is there. So there has been a small company that has been developed in order to help us to develop this device. And so it's still under development, but you got the idea that we want, hopefully before the end of 21, be able to bring that in the biolab to be able to do some freely moving experiment on mice. So next to come, because I did not show you that, is the in vivo brain activity that we are now pursuing in order to demonstrate the interest of the technology. So, okay, so I will stop now. I'm did 20 minutes, so I hope it was okay. So I have a few questions on this chat here. So from Ingrid, do you still need to remove the cortex to image activity in the hippocampus? Yes, indeed, because the endoscope head is still 2 millimeter diameter. What it brings here is that it's a 2-photon device. So okay, so depending on the microlens we are using, so the focus of the spot is between 300 micron and 600 micron in front of the distal lens of the endoscope. So if you don't remove the cortex, then you would be able to go up to, let's say, 0.5 millimeter into the brain. That would be the limit. Otherwise, you need to remove the part of the brain to go. I think that's it, no?
No, so following this question, maybe I have another question. This is concerning the tuning of the depth
Yes.
within the brain. Does it mean you can tune it after implantation, or is the tuning done during the implantation? Is this something which is...
Yes, so in this version, the tuning will be done during the implantation. So the idea, Ingrid, is to place the device on the head of the mouse, and then you start to make an image. So the head is still attached. So you see that this screw is able to hold all these mechanisms, such as the mouse cannot move. So in this case, you tune then that screw in order to look at the plane which is of interest, where you see your neurons. So you tune that. And after you detach, then the device from the, let's say, the attachment, and then the mouse starts to move freely with the device on the head. But in that case, you cannot change the imaging depth, right? In this version. There will be probably a future version when this will be possible to change the depth through electrical signal, but at least in this first version, this will be manual, and you cannot change that.
So I suppose, I mean, the goal is to have longitudinal analysis on the same animal. So how long did you manage to keep this device on the animal head, and how long did you image the same animal for?
Yes, okay, so this work has not been done by us. It was done by Cathy Ventalon using the same type of device. So I think with this device, they are able to look at the animal sessions that can be of the orders of, let's say, 1 hour, you know, with this on the head, eventually a bit longer. And then when you remove then the endoscope, the mouse keeps, let's say, this bottom interface on the head, and you can reuse the mouse again. And I think here there is no limit. So we are in the same situation than the head restraint experiments that are done in the experiment, in the biolab, means that we can keep the mouse, you know, from weeks to months, and to redo the experiments afterwards by replugging the endoscope on the head of the animal.
And concerning, my last question is concerning the activity, network activity. So the limitation seems to be the number of frames per second.
Yes. So it will be difficult to go at a larger frame rate that well, let's say 8. We can do 10 frames per second. The limitation here, and I cannot answer you now because we haven't done the experiment yet in vivo, is that you see this micro lens is obviously not as good as the bulky, very expensive microscope objective lens. So it's likely that the signal that we will get will be not as strong as with a microscope. And I cannot answer you, but it's possible that at 8 frames per second, then the signal will be not good. I mean, this is an example of 8 frames per second on a brain slice. Whether we could have this signal in vivo needs to be demonstrated.
Okay, so we have a question from Frank de Barbieux, who is asking, how heavy is the head stage?
Okay. Yes, so I think the full system here would be like a few grams, right? So we are targeting something which is like 3.54 grams for the full system.
Okay. So I don't know if we have more questions for Hervé. If we do not, we will thank you, and we are going to
Okay, so
go to the second talk. Thank you, Hervé.
Thank you very much.
So the next speaker is Thomas Chaigne, who is a researcher in the MOZAIC group. So Thomas did his PhD in Paris, and then he moved to Paris toward biology, and then he studied in Charité Hospital in Berlin. He studied he studied the directional hearing capacity in fish. So he started to work on animal models, and then now he's working as a researcher at the Fresnel Institute. And Thomas' talks will be on the photoacoustic imaging. And I think he will present his more recent development at the Fresnel Institute. So Thomas, the screen.
Yes, do you see my screen?
Not
screen?
completely. No? We see the.
You see the presenter mode.
Up screen, Thomas. Yes.
Okay. All right.
Back screen.
Okay. Now it's okay.
This is fine. All right.
This is great. Yeah.
Okay. Perfect. So hi everyone. Thanks for attending this seminar despite the hurdles. So I'm going to present you first what is photoacoustic imaging, why it could be useful for neuroscience and neurobiology, system neurobiology. And I'm going to briefly present the last result that we got at the Fresnel Institute. So here, something that you can actually see is that there is no image of any neuron here. And this just comes from the fact that there is no calcium or labeled neurons image using photoacoustic imaging, because the regime in which people usually work is more the regime in which you can image vasculature throughout many different animals or structures. And so I'm going to try to present why still it would be first attainable to do neuroscience with photoacoustic imaging, and also why it was so different so far. So as Hervé told you already, sorry, I just want to move this. There was a huge advantage of optical imaging so far, which is not only the resolution, which can be below a micrometer, but also the contrast, the variety of contrast that can give a lot of different informations to the biology. So you can have phase contrast, you can have transmission or absorption contrast, and most importantly, you have the fluorescence contrast. Sorry, just want to get my pointer. All right. The fluorescence contrast is very important. It was widely used and is still widely used because you can target genetically target specific subpopulation of cells or even structures within a cell, so that you can study really the mechanism at stake of communication between cells or of different metabolism. The issue with optical imaging is that you cannot go much deeper than a millimeter inside the tissue. And if we look at the specific example of neurobiology, we can not only, with this fluorescent labeling, you can not only look at the structure of the neurons, but you can also look at their activity throughout time. And this is really why this optical contrast is so useful. And so, as I said, you cannot go much deeper than a millimeter inside the brain. So if you look at really, let's say, classical animals that people are studying, you really have access to a tiny portion of the brain. And if you want to go and study like anything that is happening there, you rely on basically huge needles. or as also Hervé presented, you have to remove or damage at least a large portion of the brains in order to access the regions of interest. There is actually many techniques that enables you to really image non-invasively, and such a technique can be ultrasound imaging. You all know this technique. You can, for instance, look at little babies inside pregnant women, and here you can really see well the structures because you see interfaces between more or less soft or more or less hard tissues. And this really high depth comes with a fairly good resolution, but doesn't come with a very good contrast, and it's not really specific. You cannot really target population of cells with this. So we would like to have a technique that is combining the very specific contrast of optical imaging, but also yielding a nice imaging depth. And this is what can do actually photoacoustic imaging. So how does that work? You take an object, let's say an animal that contains, or a biological system that contains absorbing objects, structures like blood vessels, let's say, but it can also be neurons which are labeled with fluorescent probes. And you send a pulse laser. So in a nanosecond regime, it's different from two-photon microscopy because it's not femtosecond lasers, really nanoseconds. Light will be scattered in the tissue. You don't rely on being able to focus light down to a narrow point. And this is why you can go so much deeper than with regular microscopy. The vessels or the neurons that are labeled will absorb light and they will heat up. And upon this heating, they will tend to expand, and this will generate some sort of pop, some sort of ultrasound pulse that then will be able to propagate through the entire soft tissue and then will reach the surface. So you can measure this acoustic pulse from the outside with an ultrasonic probe. Here it's a regular probe that is also used in regular ultrasound imaging. And so you measure for each of the piezoelectric elements from this probe, you measure a time trace, which is really the pressure wave hitting the sensor. So then you feed this to a reconstruction algorithm, which yields an image like this. And this is really an optical image in the sense that you have the signal here comes from the optical absorption. So if you have no optical absorption, right, you don't have any contrast where you see darker or lighter zones, you wouldn't get any signal. So although you only detect ultrasounds, this is an optical image because you have optical contrast. But since you are detecting only ultrasounds, you can go much deeper than with regular optical microscopy. So you can get really nice images here. You can look at the scale bars over here, or here there's no scale bar. This is just color that is encoding the depth. And you can go really several millimeters deep inside the tissue. And here, for instance, what you have is the vasculature around a tumor that whose cells have been labeled with some derivative of melanin. So you have good optical absorption contrast. So first point, which is nice because this can be very specific. The depth is several millimeters. Of course, there is a small price to pay, which is the resolution is not as good as in optical microscopy. It's typically of a few tens to a few hundreds of micrometers, depending on the frequency that you are detecting. So this photoacoustic imaging technique has been now used a bit for, let's say, proof of principles for neuroscience. So here you can have, can see the images of the vasculature of the brain of a mouse. But you see, so here, this is the vertical direction. So you see that really, when you go in depth, you don't really see any small vessels, and not speaking about neurons at all. What's nice actually is that you can follow, you still have some sort of a functional signal because you can follow the oxygenation of the brain, of the vasculature, of the blood. But really the quality of the image is not good enough so that you can study any neuronal communication. There were a few actual neuroscience studies using this photoacoustic imaging technique. The nice point here was that it was really done through intact scalp and skull. So there was no surgery involved. But the resolution was limited to, was very coarse and was limited to 200 micrometers, although the depth was really much larger than with optical microscopy. But what we would like to have is really be able to do calcium imaging, but and keeping the single neuron resolution. This is really the main goal of this project. So let's look at two-photon microscopy and how it works. So in that case, you focus the excitation beam, the femtosecond laser, to a point, and then you scan this point on the object, on the neuron of interest, and the emitted fluorescence will give you enough signal to build the image. And now if you have some fluorescent indicator whose optical properties is proportional or is some sort of proxy to the membrane potential, for instance, is proportional to the calcium concentration, you can follow dynamically the activity of the neurons if you are able, of course, to image fast enough. But now if the neurons is buried deep inside a large piece of tissue, well, you cannot focus the light beam anymore. So you cannot even excite the fluorescent signal. So there's no image at all. There's no signal. It's not that you have a blurry image, it's that you really don't have any signal. But the good thing about fluorescent indicators is that they are absorbing particles, right? Like fluorescent molecules will absorb energy, lose some of this energy by wiggling a bit, and then re-emit the rest of the energy as fluorescent photons. And in fluorescence microscopy, you care about collecting this emitted fluorescence, but in photoacoustic imaging, you can use the non-radiative parts, right? So the heat that is generated by this wiggling of the molecules. And so you can probe this absorption by probing ultrasonic waves that are generated by these absorbing particles. And so if now you are able to measure the ultrasounds, you will see a change in the amplitude of these ultrasounds whenever the properties, the absorption properties of the molecules will also change with brain activity, neuronal activity. And so the detection of those ultrasounds is the whole thing here. There are a few proof of principle experiments where people do calcium imaging, again, here through intact skull and scalp with photoacoustic imaging. So the setup is fairly simple. You have an optical fiber here that goes through a transducer. So transducer is basically a collection of microphones for ultrasounds. And it eliminates the entire brain here. So this is really, the nice feature is that you really have a huge field of view. And then the transducer array will collect the ultrasound, the emitted ultrasound. You can see here, for instance, that this is really the optical absorption that is changing with the calcium concentration. For some molecules, it can be only the emission, but in the case of GChemP here, this is really the absorption. So this is why we can, with photoacoustic imaging, see some activity. So you see that we have they have some photoacoustic signal here, but the resolution is really coarse. They can go deep, but at the price of totally losing any single cell resolution. They can have some functional signal here, but studying system neuroscience here is really beyond the reach. And this is due to the fact that the piezoelectric detection of ultrasound is really limited in terms of how high the frequency can be. If we want to be able to see single neurons, so let's say having a 10 micrometer resolution, we need the 100 megahertz component of the photoacoustic signal. But if you only have piezoelectric detection, you barely see up to 50 megahertz. So we need another kind of detection in order to get this really high resolution. And for this, we developed an optical detection scheme for this ultrasound. So how does this work? You have a laser, the nanosecond laser, that is shining on the neurons and that will generate the ultrasounds. But then now you have a second laser that comes here that is scanned over the head of the animal and that will measure this acoustic signal. And the key component here is this Fabry-Perot cavity. So it's a complicated name for a very simple object. It's just a polymer layer that is sandwiched by two mirrors. Okay, so you have these two facing mirrors on top of the mouse head. And these two facing mirrors have some peculiar interference effects, which give the following results. If you change now the color, the wavelength of this incoming reading beam, the light that will be reflected by those two facing mirrors will change. The amount of light that is reflected, and if you measure with the photodiode here, will change. So there are some wavelengths which are way less reflected by this Fabry-Perot cavity. So now if you fix the wavelength of this reading beam to a wavelength which is in the middle of the linear range here of this really high slope, you can see some acoustic effect because the position of this dip over here depends on the thickness of the polymer layer. So whenever you have ultrasounds propagating through the polymer layer, it will modulate the thickness of the polymer layer, which will in turn change the position of this dip. So if you fix the wavelength here, a small change in the thickness of the polymer layer will result in a large change in the reflected light that you measure with the photodiode here. And so if you do everything right, here on the photodiode, you really listen, you're listening to the acoustic signal propagating through the polymer layer. And so this is the basic idea of the technique. Of course, this is a bit limited because you would still have a 10 micrometer resolution, but I want to show you that this could not be so bad. This is the movie that Hervé showed before. Here, it's really nice. You have a lot of details. You see the calcium signal also in the axons and the dendrites and the image, which looks really cool. But then when you look at the analysis pipeline that most neurobiologists do, they have this kind of image, then the segment, the image, and then this is the calcium trace that they extract. So basically here, what we are offering is something that you would do directly. You would have this kind of images. So of course, the pixel would be larger, and it wouldn't work if you would have really close neurons. But okay, let's say you can compare those two images if you're only at half a millimeter in the cortex. But then if you're going at 2 millimeters, this is the kind of images that you would have. So going deeper means losing a bit of resolution, but this is not so bad actually. So now a bit of preliminary results. First, those Fabry-Perot cavities, you cannot buy them, right? You have to make them. So hopefully in the Fresnel Institute, we have a whole team, a whole group that is dedicated to making this thin film coating. So we have to custom made all these Fabry-Perot cavities. And for now, we've only been looking at phantoms using 10 micrometer beads. And in the first place, we were even looking only at signals that's coming from transducers. So instead of having absorbing object, we are sending with some sort of ultrasonic speaker, some ultrasonic signal on the Fabry-Perot cavity. And we got some signal. So here, the cavity would be placed over here. So it's a bit different from what you have in microscopy, where you would have X, Y direction. Here, it's really you have X and Z axis. And then this is some first images that we got. So you see that this is really far from seeing any single neurons. But the good thing is that we really already have some signal, and now we are improving the scanning scheme and also the sensitivity, the way we make the cavity to make them much more sensitive. And we need to increase the sensitivity because the frame rate that we need to achieve is about 1 or 10 hertz. So we cannot afford to wait for several minutes like we do right now. And this is all the work of Jérémy Secours, who's a postdoc at the Fresnel Institute, working with me. Okay, thanks a lot for listening. Here are a few papers, reviews about photoacoustic imaging, and I would be super happy to answer any question. Thank you very much.
Thank you, Thomas. So I don't know if we have already some questions. Maybe I could start. Maybe if you could come back to your previous slide, showing the work of your postdoc, yes. Jérémy. So if I understand correctly, here you are imaging directly within the brain.
No brain. This is what I said. No brain. I wish.
So
this is all phantoms, or it's only through water. There is no scattering tissue. But for the acoustic problem, it's not really important because the acoustic properties of brain tissue or water are assumed to be equivalent.
Yeah, but what I don't understand. So I thought you were directly onto brain. So are you planning to start with brain slides, for example?
Yes. So yes, exactly. This is the point. Like we would go directly on brain slices, or even with we now have some brains, actually some rat head that are labeled with TD tomato. But the first thing is that we know that if it's not working really nice already with phantoms like this, so just collections of 10 micrometer bits, it would be really hard to make it work on actual animals or thin slices. So this is why we're refining this. But we are also running tests to measure how much signal do we get from labeled neurons with, for instance, GCamp or GFP or TD tomato, as I said, because those probes are really optimized for fluorescence, which actually makes it not optimal for acoustic signal generation. So we are running some tests, not using this kind of optical detection, just having a regular transducer-based detection, and we run some tests here in the lab and also at the Neuroscience Institute of Latimon.
Okay.
And we are seeing actually quite a large amount of signal. So let's say once we have a bit more sensitive cavities, we should actually see some signal.
Okay.
Not functional yet, but getting closer.
A question from Bilal El-Wali. What is the effect on the biology of brain and cells after the photoacoustic heating? And do we really could consider it as a non-invasive technique?
Yes. So the heating is about 1/10 of a degree of Kelvin. So this is really not damaging the brain at all. But of course, there is a limit. We have to make super sensitive cavities because we cannot send as much light as we want to excite those for acoustic signal. So there is definitely a limit, but within this limit, the heating is really not problematic. It's non-invasive, just like regular cortex imaging with 2-photon microscopy is. There is a surgery that we have to make in order to place a cranial window. In 2-photon microscopy, the cranial window is just a piece of glass. In our case, this would be a piece of glass or plastic plus the Fabry-Perot cavity on top of it. But we are really developing this technique to make it as convenient as possible to be used by a biologist that used to do this cranial surgery.
So that's maybe answer Lydia's question. Lydia's question is, are you planning to do it on animal immobilized or moving animals?
For now, they would be immobilized. So again, they would have a hand mount, like in 2-photon microscopy, and they would be immobilized under the microscope. Of course, one idea would be also to combine this and what Hervé has been showing previously, to have a reading system that is not a complicated microscope, but is doing it through a fiber. But this is bringing even more complications, technical. So, but of course you can imagine this. So, regarding the time frame, let's say the microscope, the scanning microscope here is working. Now the main point is really how to design how to make those cavities so that they're sensitive enough. So it's a matter of months. Then doing it through a fiber in a freely moving animal, this is a matter of years for sure. Then regarding the use of this technique in humans, so photoacoustic imaging is something that is used clinically, for instance, for breast cancer or in dermatology also, because you can look in depth inside the different layers of the skin. But this brain imaging, neuron imaging technique, you cannot apply, at least the one I showed you, in humans, first because you need this craniotomy, right? You need to remove a part of the skull and replace it by a window. So, and also, it wouldn't be so useful because you go from, let's say, 1 millimeter depth, maximum depth, to 2 to 3 millimeters, a bit more if we get super sensitive, which is a huge gain for a mouse, but for human, it's not so much. Plus, you have to label the neurons and with GCamp, which is also something you could not really do in humans. So in humans, you could imagine to go to do photoacoustic imaging with really low frequencies if you want to go through the skull, and you could look at, for instance, functional signals from the oxygenation or the blood flow, but not really like single cell resolution.
Okay. So do we have more questions for Thomas or Hervé? If we do not, we are going to thank both of you for this really inspiring presentation. And if you have any question for Thomas, Hervé, or the MOZAIC team, do not hesitate. They will be part of NeuroMarseille in the future and really happy to participate to our animation and trainings for students. So just contact them and do not hesitate to share with them your research. So I don't know, maybe Ingrid, do we have, do you want to finish, to conclude this session?
Yeah, thanks everyone. We are really sorry for the technical problem. I hope the next session will be okay. We will see you in September the 16th, I think. And this session will be in replay on our YouTube channel. So do not hesitate to go on it and watch it again, or maybe share it with your team. And thanks again for your comprehension. Have a nice day. Bye-bye.
Bye. Have a nice summer. Bye. Bye-bye.